Good morning, everybody. Let's get the class started. And this is going to be the only one lecture I'm going to deliver in this week. On Wednesday and Friday, we're going to have student-led discussions, and then next week we don't have classes. Spring break. So today we're going to continue to describe epigenetics and IPS, the induced pluripotent stem cells. So in the previous lecture, we learned that, Yamanaka started with 24 factors and then narrowed down to 4 factors, the so-called Yamanaka factors, OCT4, SOX2, KL4, and CIMIC. The 4 transcription factors can be used to reprogram many cell types toward pluripotency. So that's a major breakthrough, but at that time when it was first described, and even today, we still have quite a few concerns. At least on this slide, I will explain, the several concerns that people have over this new technology. So first of all, there are many ethic concerns. First of all, this is the cell is derived from a somatic cell source. It's not a reproduction cells. It's unnatural. So although you can claim that you will only generate iPSCs and then use the iPSC for in vitro differentiation to generate tissues, you will never do cloning. But the cloning is always a possibility. If you generate a iPSC from somatic cells, you can always use the chimeral formation route to generate an individual. So that is possible. So that's one ethic concern. The other ethic concern is it is so easy to collect patient cells. Like if you do a mouse rinse, you will get some cells out of a patient. So if the technology is now so easy, probably sometimes you forget to tell the patient before you do the experiment. You probably will just collect the cell and then reprogram the cell toward pluripotency, and then you use the cells for your research or even more advanced purpose. Then the patient's right is violated because you're cloning that patient. You're sort of cloning that patient. Although you may not generate an individual, but all the cells are kind of all replicas of the cells of that patient. Okay, so that there are a lot of ethic concerns, but other concerns are about whether this is a valid technology. Can we eventually use this in clinic healthcare? So people are worried that the epigenetic memory of the somatic cells will be kept. Like we will have examples in today's lecture. That's indeed the case. I don't know if we have completely overcome that problem, but if you do iPSC from B cells, if you do it from from fibroblast, the outcome could be different because every cell type will have their own epigenetic marks. Can you completely erase the mark and make them to bear the mark of induced pluripotent stem cells? So that's still a question. There's still a question mark there. And additionally, some of the reagents, like CIMIC, one of the Yamanaka factor, is known to be an oncogene. So it's an oncogene, it causes cancer. That means, if you have CIMIC in the mixture, then eventually, even if you differentiated the cell, maybe eventually those cells can have a higher potential to become cancer cells. So that's one concern. Additionally, when you try to deliver the 4 factors into cells, usually we use viral vectors. In Yamanaka's case, he used the retrovirus, I believe. Sometimes we use lentivirus. All these viruses have, unknown. Have proteins of unknown function. So when you use a viral vector, you're going to have carryover of the viral proteins or the viral genome. Even if it doesn't encode protein, it could encode something else, like a short RNA. You don't know whether those carryover will carry risk or not into a patient. So that's, additional concerns about the safety of using the IPSC technology. And lastly, even today, the generation of IPSCs is still a rare event. It means you start with millions of cells. If you're lucky, you may end up with maybe a dozen cells, 20 cells, and eventually you can culture the cells up because they're highly proliferative. They're like embryonic stem cells. So you can generate a lot eventually, but the efficiency, if you compare the starting point, the millions of cells, but at the beginning you only get numbers of cells, like 20, somewhere like that. So it's a rare event. The efficiency is very low. So with that, people will argue, because you cannot efficiently reprogram a lot of cells, that means a lot of random events may contribute to the generation of IPS. Because the process is sort of random, we will have a hard time to understand what's going on during the course of cell conversion from fibroblast toward a pluripotent stem cell. What's happening, at each step? We still don't completely understand the progress of iPSC generation. So today I'm going to take care of some aspects that we have just mentioned. First, I want to discuss the epigenetic memory of iPSCs. In this paper published in Nature in 2010, and this group did an interesting experiment. So they isolated embryonic stem cells indicated by FESC. That's basically that means this is from fetus, right? It's from blastocyst. Therefore, it's the natural stem cell. You can isolate that, and then you can also isolate the fibroblast from the same animal. Let's see, you can isolate the fibroblast. You can isolate blood cells as well. So basically here you isolated 3 cells out of the same donor. It's easy to characterize the embryonic stem cell because it is the gold standard of pluripotency. And now the scientists did IPS conversion. They put the Yamanaka factors into fibroblast blood and eventually convert these 2 cell types toward the pluripotency. For fibroblast, they also used the nuclear transfer as another method that get the cells converted toward the pluripotency. Therefore, you started with 3 cell types, and then through all those manipulations, you get several pluripotent stem cells, right? From fibroblast, you get ESC, nuclear transfer generated ESC. You also get pluripotent stem cells using the IPSC technology. So here, remember how they use the designation of names, to indicate what they did. FESC just means natural ESC, embryonic stem cell. The NT-ESC means it's nuclear transfer reprogrammed the fibroblast derived pluripotent stem cell. And then FIPSC means originally it was fibroblast, but now it gets converted by the Yamanaka factors. Now they're iPSC. Then BIPSC means originally it was blood cell, and now the cell is converted by Yamanaka factors. Now they're iPSCs. So all those cells are now pluripotent because you use different methods to make them to become pluripotent. And then you start to do in vitro or differentiation study in a culture vessel. So they're all embryonic stem cell like, right? Pluripotent stem cells, you can differentiate them towards different tissues. And in this study, they try to differentiate all the cell types toward the bone cells or blood cells. Apparently, when you try to derive bone cell or derive a blood cell, you'll have to use different method, right? You cannot use the same method to derive both bone and blood. So you use different culture condition to differentiate the cell toward either bone or blood. Okay, so in B, what's shown is just the evidence of pluripotency. They stained OCT4, which is a protein that's relatively specific for pluripotent stem cells. You can see no matter how you get the cells toward pluripotency, they all express OCT4. They also express NANOC, which is another transcription factor specific for pluripotent stem cells. So this B panel here just shows that the reprogramming, no matter how it was done, the reprogramming was successful. Okay, all the cells are now pluripotent stem cells. Now, on the next slide, let's study the differentiation potential of the different pluripotent stem cells. In A, what they did is differentiation of those cells toward the hematopoietic toward hematopoietic lineages. So here I have a list of the different lineages they're trying to generate: erythrocyte, macrophage, granulocyte, macrophage, granulocyte, erythrocyte, macrophage, megakaryocyte. So basically, you can observe the different cell population under microscope because they have different morphology of their colony. So those during cell differentiation, those cells will generate some colony. Those colony will bear some unique shape. The colonies bear some unique shape. From the shape, you can tell whether they're erythrocyte or other kinds of hematopoietic cell lineages. Okay, now remember this is a study of differentiation toward the blood cells. Now look at the potential of the 4 different pluripotent stem cells. We start with embryonic stem cell as shown here. So the embryonic stem cell can differentiate normally toward all the different cell types, right? As shown here, you have 3 replicate. They all have very similar potential in differentiate toward the blood cells. So if you compare the embryonic stem cell toward the nuclear transfer reprogrammed cell, you can tell they are quite comparable, right? The potential are quite comparable. But when you compare those pluripotent stem cells toward the induced pluripotent stem cell, those cells are reprogrammed by Yamanaka factors. You can see their potential, their differentiation potential are lower to start with, right? So the B cell derived IPSC, although the overall potential is lower, but still generated quite some amount of blood cells, maybe half of what you are going to see in embryonic stem cell derived blood cells. But look at the fibroblast derived IPSC, as shown in here, the second group in that presentation. The fibroblast derived IPSC almost failed to generate blood cells. The fibroblast derived IPSC, they are pluripotent. They should be able to differentiate toward any cell type by our theory, right? But in reality, what you see is the blood-derived iPSC have an easier time in differentiating toward the blood cells, while fibroblast-derived iPSC were having a difficult time to differentiate toward the blood cells. Okay, so that's one observation. And then
Sorry to interrupt, Professor, but the FIPSCs, do they have a high blood cells or low blood cells?
I'm sorry, can you ask it again?
Yeah, sure. I'm sorry, give me a minute. Okay, so the FIPSCs, do they have a high blood cells or low blood cells?
You can check there because the in A the y-axis vertical axis is calling the number per 100,000 cells. So you can see it's it did generate some, but
Okay.
it's extremely low when you compare to others, right?
Mm-hmm.
You probably if I estimated the number there, it's probably 2 or 3 cells per experiment per 100,000 cells. Where well, if you observe the other pluripotent stem cell, the number is above 20 in most
Yeah.
cases. So that's why I say the fibroblast derived the pluripotent stem cell, were not as potent, doesn't have the same potential as other pluripotent stem cells in this experiment.
Dr. Liu.
Yes.
Thank you.
Sorry for interrupting as well, but I was also wondering the sample size for BIPSC, FIPSC are like 8 and 7, and then the other ones are for embryonic stem cells, they're just 3. Is
Yes.
there that could be an issue?
I think that could be an issue purely statistically. You will feel that's an issue, but when you do the real experiment, you have to take into consideration the nature of those cells, right? The embryonic stem cell and nuclear transfer reprogrammed embryonic stem cell, they are quite uniform.
Oh, okay.
Right? They're quite uniform. So that's why you can take a smaller sample number. Well, in IPSC, the variability is much higher.
Yeah, that's why they got those results. Gotcha.
Yeah. So that's why you want to have a higher sample number. When you design the experiment, you consider the nature of those cells.
Okay, that's really cool. Thank you.
Good question, though. It's a very good question. Now let's move on to the B panel. What they are trying to do is using this alizarin red, which is specific for bone cells. So they are trying to differentiate the pluripotent stem cells toward bone. Okay. So now the result is summarized in C and D. And in C, what you can see is no matter you're using the fetal embryonic stem cell or nuclear transfer derived embryonic stem cell or fibroblast derived iPSC, you have quite a comparable degree of differentiation toward bone cells. So now it looks like the fibroblast derived stem cell have the same potential as embryonic stem cell in differentiate toward bone cell. But the blood derived iPSC cannot have the same level of differentiation compared to other cell types there. So one thing I want to mention is, the blood cell is, in terms of relationship toward bone is more remote than fibroblast. Fibroblast is closer. If you draw a differentiation tree, the fibroblast is closer toward bone, while the blood cell is kind of remote from bone. So therefore, from this experiment, they did this 2 experiments, you can sort of conclude that the cells like to differentiate toward their parental cell type. Like the blood-derived iPSC like to differentiate toward blood cells, while fibroblast-derived iPSC like to differentiate toward the fibroblast or cells more closely related to fibroblast. Okay, so indeed, they confirmed that conclusion. Basically, they use genes as markers to indicate differentiation instead of using a dye like in B. So here they studied the marker genes. As you can see, again, the ESC and the nuclear transfer derived ESC, they have a similar differentiation, very comparable. And the fibroblast derived iPSC even generated higher level of markers when you compare to ESC and nuclear transfer derived ESC. However, again, the blood-derived iPSC doesn't have the same level of efficiency of differentiation when you compare to other three cell types. So the conclusion here is what I have stated. The cell prefer to differentiate toward their parental cell type. That means they have some epigenetic memory there, that during the process of reprogramming, the marks are not completely removed. They have some memory there. And then here is from the same paper, what they did is methylation sequencing. So they studied genome-wide CPG methylation. You remember the methylation is always on a cytosine, so it's on cytosine. So they sequenced all the methylations throughout the genome, everywhere. Then you generate patterns, right? Each cell will have their methylation pattern, and then they did the statistic treatment, some method. If you're interested, you can learn by yourself. So they did that, and then they tried to cluster the cell based on how close those patterns are resembling each other. Okay, so this is so-called a hierarchical clustering. And from this result, you can see that the ES cells, you have 6 samples. The naturally derived ES cells, they are similar to each other. The nuclear transfer derived cells are kind of similar to each other, but in rare occasions, you do see that some embryonic stem cell, the naturally occurring embryonic stem cell and then the nuclear transfer derived embryonic stem cell, they are also quite similar. So the conclusion here, I would say, is that although the naturally occurring embryonic stem cell and the nuclear transfer derived embryonic stem cell, they belong to their own distinction, but they are quite similar. They're very similar to each other. But you see that the B cell derived iPSC, they cluster with each other. That one of the clone is even more remotely related to the rest. Okay, and then the fibroblast derived iPSC had their own group. They are the one that are quite different from the rest. You can see from the tiers of resemblance. So they're quite different from them. So the analysis here of the methylation pattern again tells you that they have their unique epigenetic signatures. Although during reprogramming, I believe the majority of those epigenetic marks are removed, but still they keep some epigenetic marks. So eventually, I mean, in the future, maybe we will try to come up with better strategies so that we can remove this unique marks as complete as we can, but as it stands. For now, still the cell type, the somatic cell, or the origin of the somatic cell still contributes hugely toward the quality or the characteristics of the induced pluripotent stem cells. So that's still a concern there. Sometimes you can use that to advantage. For example, like if you want to transfuse blood cells, you want to generate blood cells for some leukemia for that purpose, and maybe you can take the blood cell and generate iPSCs. Maybe you can take that as an advantage, but in most occasions we want to have a high quality, high fidelity iPSCs. So we try to remove the epigenetic mark as much as we can in most occasions. Then somebody had a question?
Yeah, I had a question.
Sure.
So you said we can use a blood cell to produce induced pluripotent stem cells for previous.
Any cell type. You can use any cell type.
Okay, but blood cell doesn't have a nucleus. How come you turn blood into induced pluripotent stem cells?
It's wrong. Only the red blood cell doesn't have a have nucleus. All the others, like lymphocyte and macrophage, they all have nucleus.
Okay, so we use those. Okay, thank
Yeah, you can. Of course, you can't use red blood cell. They don't have
Mhm.
nucleus.
Okay,
Good
thank
question.
Thank you.
Yep. Uh, since Shinya Yamanaka's work, his breakthrough, people jumped into that direction of research for us for a span of about 10 years. Many different methods of iPSC generation have been reported. And here, it's an outdated summary. I took it from a 2014 paper. These are only the major breakthroughs following Yamanaka's work. So Yamanaka described at the beginning what you can use is this octavosux2-kel4 and CIMIC. Well, the CIMIC is kind of dispensable. Without CIMIC, you still can get IPSC. It's just the efficiency is not as great. So that's why you have a dotted line to circle CIMIC. And then Thompson's group reported for human, you can use octavosux2 nanog and lin28. And then different groups, other groups reported other combinations, the combinations of transcription factors that you can use to reprogram cells. So some are quite similar to the original, like shown in row 3, 4. They are quite similar. They just replace one of the factors with a closely related one, and then they claim that the efficiency of iPSC generation was improved, but some others are quite different. Like in row 7, you have this sulfur, the power 2, ESRB, and lin28. They only share lin28 reprogram. It's very different from the formula that was originally described. So all in all, people are trying to do is trying to find a reagent that can give you higher efficiency and higher fidelity toward embryonic stem cells, and try to use transcription factors that are not associated with oncogene, like CIMIC, and eventually there are some formula like showing 30. It's showing 30. People start to come up with chemicals. So you can manipulate the cellular pathways using chemicals, small molecules. So you don't use any transcription factor. You just use some chemicals, combination of chemicals, small molecules. You can achieve the same goal. That is very good because now you don't have to introduce genes. Introduced genes are always a concern, especially if the gene have some function that you don't like the function to be present in cells. But chemicals is better defined, in my opinion, it's better defined because you always know the structure of those chemical. But then if you want to use it for therapy purpose, those chemicals will have to be approved as drug. So you'll have to study the toxicity and the other aspects of safety for those chemicals. But that's apparently a major advancement over the original Yamanaka factors. And in the rest of the lecture, I'm going to describe the mechanisms of reprogram. So why do we, we have some very successful strategy. Why do you care about the mechanism? As long as you can use it, you don't have to care about mechanism. That's some argument. But in reality, we always face challenges about mechanisms as scientists or even in industry. You always care about mechanisms. You care that more than the original discovery, than the outcome. You care about mechanism. Nowadays, if you propose to any pharmaceutical company about a prototype of a drug that you have invented, first they would like you, of course, to demonstrate it works. And then they will ask you, do you have a mechanism? They call it MOA, this mechanism of action. Do you have it? If you don't have the mechanism of action, they don't care about your discovery, your outcome, because the outcome sometimes are not that reliable. Even if you can prove it 1,000 times, it may not be true if you cannot demonstrate the mechanism. It could be still, if you don't know what's going on, the details of what's going on, it could be something, a random error that just happened again and again. But if you know the details of what's going on at each and every step, and then the likelihood of that to be an error or some other, random events is much lower. So, and when we describe the mechanism, the discussion start to become a little bit philosophical. The scientists start to think things in a very general way, like when you have a cell differentiation, let's think about embryonic stem cell differentiation toward fibroblast. So basically, we do a reversed sequence here from right to left. So an embryonic stem cell may take 5 steps from E to D to C to B to A, and then eventually to fibroblast, right? Those A through E are just different steps. So we know that the embryonic stem cell must take a very defined order of events to become fibroblast. Those events are very well defined because we're always studying the cell differentiation process. But what we don't know is, when a cell get converted from fibroblast toward pluripotency, do they still follow a reversal of the same road? That's a question scientists asked at the very beginning. So in ideal situation, you would think like the cell will just follow the reversed order of the steps and then become pluripotent stem cell. So that's a model, right? That's a model. That's a possibility there. And then others proposed that there may be other mechanisms there, such as variable sequence of key events. So you have to have this A, B, C, D, E, 5 key events, but maybe this event doesn't have to happen in the reversed order, right? As long as these 5 key events will happen, they may happen in a different order during reprogramming. So they say maybe this is, of course, just one example. They say it is possible to go through steps from B to C to E to A and then to D. Of course, other orders may still, work as well. So it's a variable sequence of key events. It's another theory there. And then the third theory there is maybe the cells will have to follow the exact steps. It's the reverse the steps of differentiation. Maybe the cells will still have to follow the same order of events, but the time latency may be different, right? So maybe it stays longer in the process from A to B, but from B to C, it just takes a much shorter time. It's a quick transition, so like that. Of course, you can have additional theories there, such as a variable sequence combined with time latency. Okay, so that of course will become more complicated, but based on this simple theoretical predictions, scientists use this as a guide to study whether it is the case, whether the reprogramming works like this. Do you have to follow an exact order, or the order is, you don't have to follow exact order? But that is just, some philosophical thinking. And here I gave you the more complicated thinking over what's described on the previous slide. So scientists proposed that the somatic cell origin also is very important. Of course, it is very important. Here they have 2 models. And one's called a deterministic model. The other is called a stochastic model. And these 2 models is further complicated by the cell origin, because in the same sample of cells, maybe some of the cells are more permissive, while the other cells are non-permissive, which is a possibility. You have 1 million cells, maybe only 5 cells are permissible. Therefore, those 5 cells are converted, while the vast majority of the other cells are not, which is highly likely, right? So you have to consider the quality of the donor cells, which apparently can contribute to the success, the rate of success for reprogramming. And then on top of that, you have this model called deterministic and stochastic. Let me explain this models. You see things are getting more and more complicated. And in this deterministic model, the scientists propose what's happening here is it's deterministic. That means if you put the factors there, define the order of events will occur. But on top of that, maybe the cells are not uniform. Some cells are more permissive, some are less permissive or not permissive. Therefore, they have this three cases. In case A, basically all the cells are permissive. Therefore, you put the factors in there, they all get converted. So you're going to have the same latency because the cells are kind of uniform. You have the factors there. That is the deterministic model in here. Therefore, everything happens almost in a synchronized fashion. You have the same latency. But you can make this model more complicated if you consider permissive versus non-permissive. In the non-permissive model, they even divided that to 2. One is predetermined, the other is called induced. You have some kind of elite cell there, permissive cell there. So you have maybe a lot of cells in there, but only some are permissive. But in this deterministic model, as long as you have the factors there, those permissive cells will get converted, while the non-permissive will never get converted. So the induced model is very similar to the model in B. Basically, they say no cell is permissive at the very beginning, but once you transfer the transcription factors in there, some permissive cells will be induced out. So maybe some cells will be more responsive to the transcription factors, and then they become permissive. Though they get converted. So remember, in this orange-colored ones, this is deterministic model. That means nothing is random. You put the factors there, define the order of events will occur. Therefore, you're going to have a same latency. But in contrast to that deterministic model, you have stochastic models. Stochastic model says, so we're still talking about these 3 permissive cases. In first case, all cells are permissive. In the other 2 cases, only small amount of cells are permissive. So I'm not going to discuss that further. I just describe what is stochastic. In a stochastic model, it says when you introduce the transcription factors in there, it's going to induce some random events. Because the events is random, the cells, of course, will experience different combinations of those events. Different combinations, and then you're going to have variable latency. Some cells will experience an easier route. Some cells will experience a more difficult route. And eventually, you see a high variability in latency. That means maybe, of course, over time, you're going to accumulate induced pluripotent stem cells, but those cells didn't go through the same route. They experienced, at least in stages, they experienced something that we will never be able to define. Right? So you put the transcription factors in there, the cell are now confused. They receive the transcription factor. They're no longer bone cells. They're originally bone cells, but by expressing those transcription factors of pluripotency, they are no longer bone cells, they're confused, and then they are making some decisions. They're trying to adapt to the new environment, and then those cells are different in their response. Eventually, some cells will come out to become iPSCs, but those we will never be able to define because the roles are different. Okay, that's what is called a stochastic model. And to make that even more complicated, in this review article, and this famous scientist Rudolf Jenisch proposed that maybe both are present during both stochastic stage and the deterministic stage are present in the course of reprogramming. Okay, so here, let me explain to you what is deterministic model. So in the x-axis is latency, of course, it's a lapse of time. And then in y-axis, that's the conversion rate. As you can see, in the deterministic model, so you put in there, because all the cells are going to have the defined steps, therefore the generation of iPSC will happen at one time, like for example, like 10 days, all of a sudden, all the cells get converted. That's the deterministic model. And in the stochastic model, because the different cell will have to experience different roads toward iPSC, you see the curve as the sigmoid curve there. Okay, it's not a straight line like at one time you generate iPSC, but rather, you accumulate some events over time. But in the other models, let me explain one of the models. It's called a biphasic model. That means maybe at the early stage of conversion, the conversion is multiple steps. At the early stage, it is deterministic. That means something will just happen following defined step. And then they will experience a period of randomness. This is the stochastic stage. Now the cells are trying to make a decision. And as a scientist, we cannot define those stages any longer. And then eventually at the end of reprogramming, the cell will resume some kind of order. And the last few steps may be defiled. They may be, for us, we probably will be able to define those stages. Okay, so at the end, it become deterministic again. But during the middle, you have some stochastic stage involved. So now here I would like to tell you what have we learned already, what we have learned. So basically some of the steps is deterministic. That means it will have to happen. It has to happen and following some kind of order. And here we take mouse embryonic fibroblast example. It will first go through intermediates and then become iPSC. Because at the beginning, they are fibroblasts. Fibroblast belongs to the category of mesenchymal cells. Mesenchymal cells, as you can see from the shape of them, they are more independent from each other. They're not in tight contact with each other. Therefore, they are more mobile. They're easily moving around. Like some of the cancer cells will gain mesenchymal ability because cancer cells are usually epithelial cells, right? But some cancer cell will gain the mesenchymal quality, so they can go to remote place. That's how cancer migrated to a different organ. That's when cancer becomes mesenchymal cells. So mesenchymal cells, they're mobile. Their shape is elongated. They don't have a cobbler or that kind of shape that mediates a cell-to-cell connection. So they are more mobile. So you start with MEF cell, mesenchymal cells. So they will have to lose their mesenchymal gene expression because eventually they become epithelial cell. iPSC is epithelial, okay? Embryonic stem cell is epithelial. So the fibroblast will have to lose their mesenchymal gene, and over time we will have to gain the epithelial gene like E-cadherin and some other epidermis genes such as keratins. So at the beginning, they lose the mesenchymal genes, and then at later stage, they will have to gain the epithelial cell genes. And then at the end, right before the complete reprogramming will happen, the cells will express genes like OCT4, SAL1, and SAL4. You can see here, OCT4 expression is right before the cells become pluripotent stem cell. While other pluripotent genes like NANOG, SOX2, those genes will express when the cells become iPSCs. So OCT4 will appear sooner than the cells actually become iPSCs. So this, studied by many scientists. These are conclusions. So some of these steps are deterministic. That means the cells will have to go through these steps. These are the defined steps. So remember this, OCT4 is activated earlier than SOX2 and NANOG. But at the end, the transgenes are abandoned and their endogenous counterparts are activated. So if you don't understand that, I can explain a little. The transgenes are the genes that you deliver into the cells. Okay, you deliver 4 factors in there. They get integrated into the genome because they are delivered by retrovirus. They get integrated into the genome, but eventually these transgenes are all inactivated by methylation. The cell will no longer use these transgenes at the end of reprogramming. Instead, they are going to activate the endogenous counterpart. Because in MAP cells, in fibroblast, those pluripotent genes, the endogenous genes are silenced because the fibroblast will not express pluripotent stem cell genes. But at the end stage, when they become induce the pluripotent stem cell, those genes, the endogenous genes, will have to be activated. So the exogenous, the one that you introduced, will be getting inactivated. So in this experiment, someone published in 2013 in Nature Cell Biology, they studied the timing of OSKM factors. So basically, in some of the experiments, they give OKMS, that's the 4 factors, simultaneously from the very beginning. But in some experiments, they give OK first, OCT4 and KOF first, and then followed by a combination of OCT4, KOF, and MYC, and then followed by MS, which is MYC and SOX2, and eventually they give SOX2. Why? That is because in this model that people have demonstrated numerous times, SOX2 appear much later than OCT4. Therefore, maybe SOX2 is not required at the early stage of reprogramming. That's why they come up with this idea to give those 4 factors at different time. Let's observe the result. As you can see, the y-axis is the efficiency of cell generation, and the first column is OKMS. That's the original formula. That's the reference point. As you can see, if you omit CIMIC, you're going to have lower reprogramming rate. But if you follow the new timing, the new schedule, so you can see you have a huge improvement. So you give OK first, and then followed by M, and then followed by S. The SOX2 was given at the very end, and then you have increased reprogramming efficiency. So now they start to appreciate how the different factors contribute to how they contribute to reprogramming. And it turned out that if you give the new schedule, that is SOX2 was given at the very end. So what you will see is that the expression of E-cadherin, which is the epithelial marker, is much slower, right? In the original, you see a high expression of E-cadherin in just 6 days. And then at 9 days, the level is higher. The original formula gives you a higher E-cadherin expression level. And then if you study slug, which is a mesenchymal gene marker, okay, in this case, you can see the original turned down that mesenchymal gene much faster, while the new schedule turned down that mesenchymal gene much slower. Okay, this is very interesting because what you're doing is, let's go back here. So the cell will have to lose the mesenchymal gene and gain epithelial gene. Okay, that's something that will have to happen. But in this experiment, the scientist has demonstrated if you slow down that process, basically you make that process to experience a longer time, actually you can improve reprogramming. That means the original formula was too fast in getting the cells to lose the mesenchymal marker and gain epithelial marker. Okay, we almost come to the end. I would like to tell you that there are evidences of stochastic phases. Largely, these evidences are from single cell study, because a challenge there is when we're trying to gain mechanistic insight of reprogramming. At the beginning, we're all dealing with the cells, the population of the cell as a sample. As we discussed earlier, maybe 5 permissive cells eventually get reprogrammed. So that's really rare events, but when you take the sample, you take the million of cells, the billions of cells as your sample, because that's where you start with. Therefore, you're never studying the real mechanism. You're studying the kind of unrelated or random events there. That's why, if you don't use single cell study, the conclusion is really not reliable. If indeed it is the case that only small number of cells will be permissive and eventually get reprogrammed, especially when different cells will experience different events in a stochastic manner. If you don't use single cell study, you will not gain a picture because the overall picture is misleading. So that's indeed the case, because with single cell technologies, we found that in the first 6 days after factor induction, there was high variation among cells in expression of the genes. So there's high variation in different cells. It's not like all the cells will behave the same way. They actually behave very differently. But some things are defined. You can define them, like expression of genes that control mesenchymal to epithelial transition met. So those will have to occur. So something are there that you can actually define. So cell proliferation and metabolism will have to change. We know that population-wide something will have to happen. But what we don't know is something, that occurred very randomly. And since we take a big sample, like millions of cells are studied, but only maybe 10 cells those behave differently are actually the key events there. It is very hard to study. It's very hard to capture those cells because you don't know which cell to capture. But with the single cell study, that is now becoming possible. So let me summarize the knowledge that we know for now, that at the very beginning of reprogramming, the cells will have to increase proliferation. They will have to have metabolic change. They will have to initiate mesenchymal to epithelial transition. These things can be defined. But then the cell will go through a stochastic phase, so including a rate limiting step. Basically, all the cells will experience the same change, such as increase the proliferation, metabolic change, but only some cells, we don't know which cells, they are going to have some kind of stochastic events going on. Like in them, they are trying to adapt to the environment. They are trying to remove some epigenetic marks. They're trying to open up some gene expression, but in different cells, that happens in a different way. So that's why those cells will experience a stage in which we cannot define what's going on. But eventually, when the cells come out of that stage, they will resume some order. Like they may start to express octa4, and then eventually they're going to express nanog. Scientists observe that once the cells start to express nanog or even SOX2, and now the cells are more and more uniform. They are more and more like each other. Okay, so at the beginning, fibroblasts are very like each other. If you do a single cell sequencing, you know the heterogeneity is not high. At the end, the heterogeneity is not high. But at some stage during reprogramming, the cells are very different from each other. So I guess our task is to capture those cells that eventually will become IPSCs and study what's going on in those cells. So try to define the stochastic stage, but maybe that's an impossible task, because if that stage is truly stochastic, that means the cells may have thousands of ways to go in that stage. So that to define it is meaningless, because the cells won't go through one way, they could go through many ways. Okay, I guess this lecture is pretty difficult, but as I said, the study the details of mechanisms is super important, not only in IPS generation, but also in other scientific studies as well. I hope this is, although difficult, but kind of it promotes your thinking. Okay, that's the end of today's lecture. And thank you. If you have a question, you can stay behind and ask me.